In this experiment, we will know how to extract a DNA and what are the chemical principles behind all of these. A DNA is a special molecule that can be found in all living organisms on Earth. Part 1. Here are the materials. Procedures. First, we will be using Ziploc bag to crush the bananas. The purpose of this is to physically and mechanically disrupt the cell wall of the individual banana cells because bananas are plants and they have cell walls that have to be broken down before the DNA can be released from inside the cells. Second, we will now prepare the precipitation solutions for this experiment. The components of this includes the table salt, liquid soap, and hot water. The role of a table salt is to neutralize the negative charges on the DNA because DNA is composed of a lot of phosphates on its backbone. It is negatively charged and the sodium ion in table salt will agglomerate or gather the DNA particles or molecules that we are aiming to extract and separate later on. For this soap, its purpose is to break down the cell and nuclear membranes of the cells of the banana that we have already disrupted because of the mechanical actions we performed a while ago. Soap helps break down lipids and proteins in the cells and nuclear membrane. Hot water will allow the temperature of the solutions to be raised and this will promote the denaturations of the proteins that are present in the cells and nuclear membranes of the banana cells. All of this will be combined to be able to release DNA from the insides of the cells. Third, mix the precipitating solutions into the mashed banana and filter it using a strainer. Fourth, we will now be adding cold isopropyl alcohol to our remaining solution. The purpose of using cold alcohol is because the DNA in banana is not soluble in cold isopropyl alcohol. Therefore, the DNA will clump or agglomerate when in the cold alcohol. Here's the result of our first experiment. In our Part 2 experiment, we will extract a DNA from chick cells. The process is quite similar to what we have done in the previous seconds of this video. The only difference is that we will gonna use here our own saliva as our main solution. Here are the materials for this Part 2 experiment. Procedures. First, prepare the salt solution. Second, gargle the prepared salt solutions for 45 seconds so that your chick cells will mix with it. Next, add a liquid soap and a cold isopropyl and gently stir the mixture and wait for the fibrous DNA to form. Here's the results in our second experiment. Summary of the chemical principles. Thank you for watching.